Welcome once more to IISER Pune. IISER Pune is a premier research institute of this country, and we of course do scientific research. We teach at the level of B.Sc, M.Sc and PhD, but we also engage with students from schools all over Pune and in fact from all over India. And as part of our outreach activities, one of the things that we have been doing for the last 4 or 5 years is organizing molecular biology workshops for school students, college students, and master's students and PhD students. Now these workshops are a bit advanced, and that is why one of the things that we have been thinking for a long time is that we are not reaching out to students who are, let's say, below class 9, 6th, 7th, 8th, and that age group. And that's why we had a chat with Science Activity Center headed by Ashok Rupner, and Ashok said that we can do that. So we had lots of discussions, and what is going to happen today in front of you is the result of that discussion. And we have put together the whole thing in the form of a demo workshop, demonstration workshop. We'll conduct lots of experiments right here, as you can see, and hopefully we'll tell you about the backdrop, the use, and so on and so forth. Okay. Now, for this workshop, there are 2 instructors who is going to talk to you about. So this is Sneha, Dr. Sneha Bhogale, and that is Poornima Raveendran. And these 2 people, they have enormous amount of expertise in doing both molecular biology and biochemistry. And they have been, in doing these molecular biology workshops since 2019. So what we are going to do, the experiments that we are going to show you, I don't think you would have seen them before. And yeah, I don't think I want to stand between you and the thing. And over to you, Sneha.
Good morning and welcome everybody. So we are going to present a demonstration session, and the title is Proteins, the Wonder Molecules of Life. So before we start with any explanation kind of a thing, we are going to do a fun experiment. Yeah? We are going to use potatoes.
Potatoes?
Everybody likes potatoes, right? Especially in this monsoon season, vada pav, bhaji, right? So today, rather than cooking with potatoes, we are going to do something fun. Are you guys ready to see what we are going to do? Yes. Great. So Poornima, over to you.
Thank you, Sneha. So, potato, everybody knows a potato, right? Normal potato, the one that you bring at home. We have taken the juice of that potato. Can you all see this in the flask? Yes. We have put the potato, cut it into pieces, put it into a mixer, ground it nicely, and you see this juice that is coming out of the potato. Okay. Now I am going to take some amount of this juice of potato in this measuring cylinder. Okay. So I am pouring some of this juice, potato juice, in this measuring cylinder. And now I am going to add a chemical called as hydrogen peroxide. Okay, this is a hydrogen peroxide is basically bleach that you use for your hair coloring, etc., or your mother might be using for her hair color, etc. This is also available in the medical shop. This is used to clean wounds. When you have any kind of wound, it will hurt a lot, but it is used to clean wounds. Okay, so I am going to add a little bit of this hydrogen peroxide chemical. If I can open this bottle, yeah. Okay, so I'm going to add this hydrogen peroxide, and now just observe what is happening here. Can you all see this at the back especially? No? Okay. Can you see this now? Yes. Wow. Okay, so what exactly happened here? I just took this potato extract and I just added some hydrogen peroxide into it. And this is the reaction. This is called as a reaction, right? This is a reaction that happened inside this measuring cylinder.
What do you think just happened? Who is responsible for this chemical reaction? Any guesses? Yes? Correct, but what happened to the hydrogen peroxide chemical? Okay, so he's saying that H2O2 reacted with the potato extract and it burst, right? Who made that happen?
The starch present in the potato.
So she's saying the starch present in the potato must be doing this reaction. That is not the right answer, but good try. Anybody wants to try?
The proteins.
Bingo. He said the proteins are making this happen. So let's see what are proteins. So right from simple single-celled organisms like bacteria, paramecium, viruses, to slightly complex organisms like fungi, yeast, algae, to even more complex organisms, plants, animals, all the living forms contain proteins. All living forms are made up of 4 macromolecules: carbohydrates, proteins, nucleic acids like DNA, RNA, and lipids. And today we are going to focus on proteins. Okay? We have eggs, a very good source of protein. We have sprouts, a very extremely good source of protein. We have milk, right? We all eat these food components. We get our protein intake. Our body uses it for energy, and it also makes new protein in the body, like hair. Our hair contains a lot of protein, especially keratin. And even our muscles have a lot of protein, right? So these are our common sources of proteins. But now, let's see what do they do. So what are the functions that the proteins do? Let's stick to our human body, okay? So, when we get hungry, our blood glucose drops down. When we eat food, the blood glucose is restored. So there are hormones, especially insulin, that takes care of this blood glucose level, and all hormones are proteins. When we fall sick, right, our immune system protects us. It tries to fight off infections, and a very important part of the immune system are antibodies. Antibodies are proteins. The hemoglobin in our blood, what does it do? It transports oxygen throughout our body. So hemoglobin is a protein. So protein also carry out the function of transport. When you eat food, the food is broken down, right? We digest the food. So right in our saliva and in our gut, we have a lot of enzymes which carry out this reaction, where they break down the food into easier, simpler components so that our body can absorb it, right? Like you have digestive enzymes, we have many other enzymes in our body that do all these different chemical reactions. So all enzymes are proteins. Some proteins do the function of storage. Like this protein here is storing iron, and the name of that protein is ferritin. Some proteins also do structural function. For example, the hair, the nails, the bones, we have proteins in them that give structure to our body, right? Receptors, this is another function that proteins do. Many receptors are proteins. For example, the light receptor in our eye, because of which we can see, this light receptor called as rhodopsin is also a protein. And our muscles, we jump, we run, we work, right? All this is because muscles are rich in proteins, and proteins carry out all these different functions. So this is just a snapshot of what all functions proteins do in our body. Now you can imagine proteins are present everywhere, all living forms, what all different functions they might be doing, right? So I just want to make you understand that proteins are present in all living organisms, and they are very important because they carry out a variety of different functions. So let's answer the question. Let's go back to our reaction where we used the potato extract. We added H2O2 and made it bubble, right? It literally overflowed. We have lots of bubbles. You guys answered the question that proteins are making this reaction happen. So let's talk about which protein made this reaction happen. So the protein that was responsible for this reaction is an enzyme, and the name of this enzyme is catalase. So what does it do? How did we get bubbles in the first place? The catalase enzyme, it breaks down the hydrogen peroxide into water and oxygen, and the bubbles you see are literally oxygen bubbles. So the catalase enzyme is the protein here in our reaction, which is breaking down the hydrogen peroxide and giving rise to water and oxygen. It is making this reaction happen, right? So now we are going to repeat the same reaction, potato extract, H2O2, everything same, but now we are going to change a few conditions. Everybody is ready for this? Let's see what happens.
This time we will ask 2 people or 3 people to come ahead, volunteers, one from every row. Can you, is there space ahead? Yeah, come. So I just want you to pour the extract and do it yourself. So take it in this small cylinder, it's the same thing. So just pour it, take it up to this 5 ml mark, and then 10 ml you will add the H2O2. Okay. Keep it. Rakh do. Aa jayega. So this is going to be slower. Yes, next person can come. Take a little bit of the extract. Next person can till 5. Can you see the 5 ml mark? Yeah. So I'll add the, yeah. You can keep it down. So this is now proof that even if you do it, you're getting the reaction, correct? It's still coming, the bubbles are here. Okay, so now what we will do is, we are doing 2 different conditions. Okay, the last person can also come and come here, take the tubes. This is the other. Okay, so these are the 4 reaction tubes. Now what we will do, we are keeping some of this mixture in this. Yeah, so you can see that the first reaction has already come out, whereas the last reaction is still happening, correct? Anushka's reaction was last, so can you see it's still taking time. And what was your name? Prajwali's reaction was the first, so you can see that it has already come out, right? Now what we are going to do is, we are going to heat the extract. So I have put this in this water bath, okay, and we are going to just keep it, it's at 70 degrees, it's set at 70 degrees, dry bath, okay, it's set at 70 degrees. I have put the solution in this dry bath, and then we also have extract on ice. So what is this? This is chilled extract, and that is going to be the hot extract. Potato extract only, but this is chilled, kept on ice, and this is old, which is why you can see it is a little blackish. This is fresh extract, which I have put in the boiling bath. So let me just add now the cold one. See what is happening, okay? Same thing, I will add the extract and add H2O2 into the extract. Volumes I am keeping constant. Last time also I had taken 20 ml. And I am adding H2O2 from this bigger bottle now. Now see what is happening when it is cold and also old. Can you see? What happened? How come? But how come? Less protein, suddenly the protein went down? It's the same extract, no? The same extract that we had, which you used. Temperature. Okay, there is a suitable temperature. Okay, let's see. So this one is now cold extract, and now the one which is kept for boiling, we will take it out in a few minutes. 2-3 minutes, and we will take this out, and then again we will do the same thing. So let me just pour the acid, the H2O2. Okay, I'll keep the H2O2 poured so that I can just add the boiled solution into the mixture. We will now take this out. ये tubes में है ये, तो अभी ये tubes निकाल के मैं और स्नेहा अभी इसमें pour करेंगे. एक एक करके सारे ट्यूब जो है इसमें डाल देंगे, ठीक है. ऑलरेडी इसमें H2O2 है. Can you see over here now? This is boiled, this is chilled and old, okay, and this was my fresh extract in the center. This is still there, bubbles coming out. So somebody correctly said that temperature is playing a role over here. Okay, this was the first tube. This was the one which is old. Can you see here? There is bubbling, but look at the amount of bubbles that are there, and look at the amount of bubbles that are there in this one, right? And the last one is this boiled one. इसमें भी थोड़े से bubbles हैं, but बहुत ज़्यादा bubbles नहीं है. दिख रहा है? So the next one is again a different demo. This is one which is over. Now, do all of you know about pH? What is pH? Here there is a pH scale that is given. This is basically a scale from 0 to 14, right? pH is a scale. You are measuring how much hydrogen is present, and according to the level of hydrogen, you can decide whether it is acidic or whether it is alkaline or basic. Same thing. Alkaline, basic, same thing. So you have 0 to 14, and 7 is called as a neutral pH. Like water, we drink water, right? What do we say? The water has neutral pH, 7, which is good for us, for our body. So you have 7, which is neutral pH. Acidic and alkaline are at the two extreme ends. Now how do you test pH? In your lab, school lab, have you tested pH? Indicators, very good. There are pH indicators like, सबसे common one is litmus paper. You must have used litmus paper, or you must have heard about litmus paper. So now what we have done is, we have two extracts in which, because of safety reasons, I have already added the acid and base. People behind, pay attention. I have already added acid and base to the potato extract. Again, same thing. I have changed the pH of the potato extract. So what was the pH of the normal potato extract? Okay, let me show you with the camera maybe, so that it's easier for you to see, especially at the back. Okay, so this is a pH paper. Okay, I will put this in the potato extract and I'll show you the pH of the potato extract. So how do we check the pH? There is a color indicator. Okay, I put it in the potato extract. There is a color indicator here. Can you see visible? So neutral, somewhere close to 5, 6, somewhere between 5 and 6. Close to neutral, not exactly 7, but it is close to 5 and 6. Okay, visible? Okay, so this was our normal pH. Now let's look at the pH of the acidic solution and quickly look at the pH of the basic solution. So I'll take one more pH paper. First let's look at acidic. Everybody likes acid, I think. First people said that pH is acid, measuring acid. Oh wow, look at that. Visible? Color is visible. So what is this now? Orangish, closer to less than 5 for sure, right? It is closer to 3 or 1 or something like that, in between that. That seems like it, right? But this is acidic pH. And now let's go to the next one, which is basic. So where I have added a base. Detergent. What is base? Soap. You use soap, right? Look at that. Oh wow. What happened? Green, blue. Here. Can you see this now? Yes, 9, 10. Okay. So that means I have changed the pH of these two solutions by adding acid and base. Now let's see again the reaction, if there is anything that is happening. I am again going to add 20 ml of this in one tube, in one measuring cylinder, and 20 ml in the other measuring cylinder. Yeah, I'll keep it over. Okay, this, and now I will add the H2O2. Okay. Is that visible now? Behind? Same. Any difference? After adding H2O2? No difference. So the first time we added, it came like boom. Even when students came in the front and they added, it still came up. The frothing came nicely. But now there is absolutely no froth here. So acid base, again, what has happened? Something has happened. What was the enzyme that we put, what was there in the extract? Catalase. Something has happened to catalase. Okay, now let's see what exactly happened to catalase.
Let's rewind a little. We added H2O2 to potato extract and it just bubbled, right? Somebody said the word burst. It just burst, right? We changed the temperature. We increased the temperature. Did the reaction happen? No. When we reduced the temperature, did the reaction happen? No, or there was a little slight weak, no reaction? Okay. When we changed the pH, when we made it acidic or basic, did the reaction happen? No. So what we are looking at right now is a simple qualitative yes or no answer. That is the assays we did for our enzyme catalase. So here at IISER, we conduct hands-on molecular biology workshops, many of them, of which one focuses on catalase. And students not only get to do hands-on experiments on the qualitative yes-no aspect of catalase, but they also work with the quantitative part of catalase where they measure how much activity is actually there, if there is any. And they also work with the catalase gene from the potato and they do certain molecular biology experiments themselves. So moving on, what changed? Right, we saw the things are not working. So what happened? What happened to our catalase that it suddenly stopped working? Initially it was bursting oxygen bubbles out and now it is not giving us any result. So let's take another example. You guys know how to cook eggs, right? You guys also know that the yellow part of the egg is the fat-rich component and the white part is the protein-rich component, right? So we are going to focus on the protein-rich component. What do? You take a frying pan, you break the egg open on it, you switch on the gas or the stove, and the temperature of the frying pan starts increasing. As the temperature increases, can you see the change in the protein now? Initially, it was liquid consistency and colorless, but as the temperature rises, the protein gets cooked, meaning the protein now loses its consistency and the consistency changes to solid and the color also changes to white. Everybody knows this? So what is happening here is also very similar to what happened to our catalase. We also increased the temperature of our catalase at 70 degrees with a bunch of other different parameters, right? So let's see what happened. Let's focus on the left side of your slide, okay? Focus on this properly folded proteins. So if we look at the protein structure, proteins are long chains, okay? They are made up of these small building blocks and they are arranged in long chains. Some proteins might have a single chain, some proteins might have multiple chains. What do these chains do? These chains fold in a certain manner, giving a proper structure to the protein, and such properly folded protein with a proper structure is functional, which means this properly folded protein is functional, it can do its job. But the moment you expose these proteins to different changes or different parameters like pH change, temperature change, salt concentration change or any other parameters, the protein structure starts crumbling down. The chains start unfolding, the protein loses its structure, and finally the protein breaks down. So such a broken protein is called as denatured protein and the process is called as denaturation. Now tell me, if something breaks down, will it work? No, right? So that is what happened with our catalase as well. At such different temperatures, high and low, and at such different pH, high and low, our catalase enzyme broke down and hence it could not do the same reaction which it could do before. Everybody understands this? So the point that I'm trying to highlight is when you change the surrounding parameters or when you change the surrounding conditions, protein structure gets affected. Therefore, protein function also gets affected, right? Now I want you guys to think on this point and tell me, what does this next slide tell you? What does it show? So what I want to drive home is that Earth, the temperature on Earth is getting warmer and warmer. The ice at the north and the north and south poles, the ice is melting. We have a lot of intense and unpredictable cyclones, forest fires, rainfalls. Seasons are changing, they are becoming drastic and extremely random. And all these things are affecting the soil, making the soil alkaline, making the soil salty or saline, basically making the soil infertile, because of which plants production is getting affected. The entire environment is also affecting the organisms that live on this earth. Everybody understands now where I'm trying to go? The moment you change your environmental parameters, proteins are going to get affected. Because we just saw how parameters control protein function.
I wanted to ask that the plants that grow around, the vegetables we eat, they have those proteins. So now, do the proteins decrease in those vegetables and fruits that we eat because of this?
So yes, there are studies which clearly show how climate change is affecting the nutritional value of the plants, fruits and vegetables that we eat, what they were before and what they are now. So yes, it might not happen that temperature increase ho gaya, protein degrade ho gaya, but it might reduce its function, which automatically reduces the nutritional value or something of that sort. But it does affect the nutritional value. We'll move to the next component. Yeah.
One more thing is that when you cook, that time also protein gets broken, right? It is so that digestion is easy. That's why we cook up protein and then eat it. If you remember, you're not supposed to eat uncooked pulses. They say, पेट में दर्द हो जाएगा. You will have a stomach ache. Why? Because protein is complex. It is complicated. If you don't break it down, that's why cooking, heating is necessary, but to a certain limit. When the plant is undergoing very high climate change and very high temperature change difference, that is going to create problem with the nutrition. Amount of protein itself is less. So breaking down is a different thing, but amount itself is less. That will become a problem, right? So there are two different things. Okay, thank you for that question. So अभी हमने क्या देखा? हमने देखा प्रोटीन का टेम्परेचर रिलेटेड क्या होता है, करेक्ट? अब हम देखेंगे थोड़ा सा यूज, कहाँ यूज होता है प्रोटीन का. बॉडी में तो होता ही है, करेक्ट? बॉडी में क्या यूज होता है प्रोटीन का? बिल्डिंग व्हाट? Muscles, hair, right? All of that needs proteins. Our organs also need protein. Cells, inside the cells there are a lot of protein. So everywhere there is protein, right? So there are lots of uses of proteins that are there. But today we are going to concentrate on health, medicine and health, okay? That is the main area that we are going to focus on, because now you've understood how important proteins are. सब जगह पे प्रोटीन का नेसेसिटी है, इट्स इम्पॉर्टेंट. तो आप मेडिसिन एंड हेल्थ केयर में सबसे पहले, सो व्हाट हैपेंड मेनी इयर्स अगो इन द 1600s, okay, people tried to, somebody had blood loss, a small child had blood loss. So one of the doctors at that time in 1600 in France, he decided, let me try to put some sheep blood into this boy, okay, because the boy's life had to be saved. There was a lot of blood loss. When they put the sheep blood into this boy, miraculously the boy survived. Okay, he had some temperature, some issues, but he survived. The second time, the same thing happened for another older man, and they tried to again put, I think, calf blood or something like that into this person's body, and they figured out that that person was not able to survive. The person died. So now this was puzzling. The person who did the first experiment was the same person who did the second experiment, okay, the Frenchman. But he was arrested because he killed a person by mistake. I mean, he was not trying to, he was trying to save the life. But instead of that, that person died. So people started wondering, how can this happen, right? Same blood, sheep ka blood, ek bacche ko diya, the bachcha survived, but then when it was given to a older man, he could not survive. Guzar gaya wo. So then what they tried to do was try to figure out. Scientists started thinking what exactly is happening. In 1800s, there was another problem. That time, in those 1800 times, women used to undergo labor, pregnancy. After pregnancy, they used to have internal bleeding. So to stop this internal bleeding was very difficult. So women used to die because of this internal bleeding. So one doctor named Blundell, James Blundell, he was in Europe, in England, okay. What he did, can you see this picture? What is this picture showing you? There's one person standing over here and there's a woman. She's just delivered a baby and she's almost unconscious, almost, because she's had a lot of blood loss. So what this person is doing, can you see over here? What are they doing? Pushing the blood, correct? Transfusion. So this is the first experiment of blood transfusion in the 1800s, 1818 by James Blundell, where this person is actually giving the blood and the blood is going down into this other lady's hand, into the veins basically. And like this again, a lot of women survived. A lot of lives were saved because of this experiment of James Blundell. But still, people were cautious because they didn't know when somebody would survive and when there would be a death. So scientists started asking this question, why is this happening? Finally, in 1900, okay, there was a scientist called Karl Landsteiner. He discovered that our human blood can be divided into 4 groups. Like you guys are sitting in 4 different lanes, 4 groups. Like that, human blood is also divided into 4 groups. You all know the groups? Now we know. A, B, AB, and O. Everybody knows this. And also there is a factor called as positive or negative. Now we know this, but that time nobody knew. When Karl Landsteiner discovered this, he won the Nobel Prize, which is considered as the highest achievement in science, right? So he was awarded the Nobel Prize in 1930 for this. So that is the earliest life-saving technology. Now what we are going to do, we are actually going to give blood right now. Okay? So we will do a demonstration for you people. We will put drops of blood. Don't worry. We have had samosa with you guys in the morning. We won't faint. We are going to give only 3 drops of blood. Okay? We'll mix it with anti-sera which we have. It is kept on ice over there. And we will see what happens. Has anyone seen how to check blood group? Anybody knows? No? Dekho, abhi dekho. Can you see the people sitting at the back? Please focus on the 2 screens. Can you see the slide over there?
Okay, so let's start with the blood grouping thing. So what I'm going to do is, I'm going to prick my finger. So I just I'll swab my finger with 70% alcohol. So I'll clean it, I'll disinfect my finger. Then I have a lancet here which has a tiny needle, a sterile needle. And I'm going to prick myself. Poornima, I'm doing the same. So quickly, in these 3 drops of blood, in these 3 drops of blood, we are going to add certain solutions. What these solutions are, we will discuss them in a minute. So I am taking a blue solution, 50 microliters, and I am adding in both the samples. Then there is another yellow. and there is a third solution, colorless. So as you notice, after adding the solutions, Poornima ma'am is mixing them.
Now we will wait, okay, because सब्र का फल मीठा होता है, ऐसे बोलते हैं ना. So अभी थोड़ा रुकना पड़ेगा. We'll wait for some time before we can see the result. Alright, let's go ahead. So now when people discovered that there are 4 different blood groups, what did they do? अभी मेरा कुछ blood group है. If I need blood, immediately from where I'm going to get blood, right? It might be, ऐसा हो सकता है कि there is nobody around me of the same blood group, possible है. So then what will we do? Therefore people started donating blood. So you must have heard of blood donation camps, and then there are blood banks. Like you have a bank for money, there is also blood bank where all the different blood groups, blood is deposited and stored properly at the right temperature, correct? Alright, so what is present in this blood group which is causing this difference? What is there in my body which is different, in my blood which is different from Sneha's blood? So what is it? There is something called as immunity, right? Our bodies have immunities. We are able to fight against different organisms, bacteria, viruses, etc., pathogens of all kinds, because we have some immunity. We have immune response. What is this? Does anybody know what is an immune response in my body? When I get an infection, what is an immune response? Basically, my body is producing proteins as a defense mechanism. So these proteins are called as antibodies, okay? These are the defense proteins of every body. Every body has antibodies, which are the defense proteins. They are the first line of defense in our body. So what happens is there is something which is foreign object. Don't worry about the sphere, it's just an example. This is a foreign object. This can be anything. It can be virus, it can be bacteria, it can be a worm, it can be anything which is foreign to my body, anything, okay? My body will produce an antibody, which is a protein. So the protein, this protein is shaped like a Y. It has 2 hands and 1 center body, okay? It has a Y-shaped structure. So this is an antibody, which is a protein, and it will react with this antigen. And what will happen is it will form this kind of a group. It will form a group like this. The antibody and antigen will react with each other. This is what gives us immunity. You will ask how this is giving immunity. So you have the white blood cell in your body. Where is white blood cell present? In blood, correct? White blood is present in the blood. Blood में से यहाँ पर antibody produce कर रहा है, मेरा white blood cell antibody produce कर रहा है. बैक्टीरिया के ऊपर what you see on top, that is the antigen. छोटा छोटा दिख रहा है बैक्टीरिया के सरफेस पे. ये कुछ भी हो सकता है, प्रोटीन हो सकता है, कार्बोहाइड्रेट हो सकता है, फैट, लिपिड हो सकता है, कुछ भी हो सकता है. सो यहाँ पर ये एंटीबॉडी इसको आइडेंटिफाई करेगा और ये वाइट ब्लड सेल जो है आइडेंटिफाई करने के बाद उसको दूसरा एक सेल होता है जो उसको खा लेगा. सो बेसिकली बैक्टीरिया को वहाँ पे डिस्ट्रॉय हो जाएगा वो बैक्टीरिया. ओके, सो दिस इज हाउ द वाइट ब्लड सेल्स इन आवर बॉडी इन शॉर्ट, ओके, इन वेरी सिंप्लीफाइड मैनर, दिस इज हाउ इट डज द वर्क. सो नाउ how does ABO blood typing work? My blood cell has got certain antigens on the surface. Can you see this round yellow things? Those are the antigens present on my blood. The blood that I gave, uske surface pe ye antigens hai. When I add the correct antibody, what should happen now? What should happen when I add the antibody? What will happen? They will come together. Like this, they will form a clump. They will clump together. एक साथ आ जाएंगे वो. This is called as agglutination. इस process को agglutination बोलते हैं. Don't worry about the technical term. The point is that they will come together like this. Now let's have a look at our slide then. If they have come together like that or not, correct? We have added blood. We have added antibody. The colored solutions you were asking. Somebody here was asking, right? What were the 3 colored solutions? They were basically antibodies. 3 different antibodies. One was blue in color, one was yellow in color, and one was white in color, for you to understand. इसलिए color difference बता रही हूँ मैं. So now look at the slide. Can you see? Is it visible? What has happened here and here? These two. What has happened here and here? Colors छोड़ दो, अभी colors छोड़ दो. यहाँ पे क्या दिख रहा है? Red color जो दिख रहा है. What has happened to the red color? Mix नहीं हुआ है? तो क्या हुआ है वहाँ पे? ये क्या हुआ है यहाँ पर? वो mix हुआ है. अच्छा. So I am getting one answer कि यहाँ पे mix हुआ है blood और यहाँ पे mix नहीं हुआ है. दिख रहा है क्या ऐसा? Can you guys see it like that? Visible? अब दिख रहा है clear? हाँ. So क्या हुआ यहाँ पे? These four are different. These three are different from this three. ये तीन कुछ अलग है और ये तीन कुछ अलग है. Yes, everybody agrees? Yes. Backbenchers, girls over here, agree? Yes or no? Yes. Okay. So what has happened over here? Now we just discussed that antigen and antibody will come together and they will group, they will clump together. This is the clumping that you are seeing over here. These three are clumped, whereas these three are not clumped. This is as it is. They are remaining two different liquids. Blood is remaining like this red in color and the yellow liquid is around it. Here also the blood is remaining as it is and the blue liquid is around it. Here this is remaining as it is and the white liquid is around it. Correct? So now we have to determine our blood group based on this. कैसे करेंगे? कर सकते हैं? नहीं पता? How will we decide कि blood group ये कौन ये? This is one blood and this is the other person's blood. Okay? I know which is whose, but we'll discuss that. So how do we know the blood grouping now? Based on? Based on the clotting. Okay, she's calling this as clotting. No problem. This is actually clumping. It's not exactly clotting, but okay. Based on the clumping that happened. What exactly happened over here? So this was the earlier slide. Correct? Remember this? When we did the three blood groups, first one, what happened? There was one group which was showing clumping and the other one which is not showing clumping. So we added blue liquid and we added yellow liquid. जिसमें yellow liquid में clumping दिखा, that becomes blood type B. Okay, then there was another one जिसमें दोनों में there was no blue and yellow. Both of the liquids were showing no clumping. Remember, red was separate and blue was separate. So ये हो गया blood type O group. Okay, and then you have two more types. One is when there is clumping in blue color. In the blue color, that time it becomes A. And when there is no clumping in B, then it becomes, that is again A. And here you have AB where there is clumping in both. Okay, let's go back to the slide again and let's just quickly see. हाँ, उसमें देखते हैं अभी तुम लोग बताओ. There's a third thing that is positive negative that I have not discussed so far. But let's just look at the blue and yellow color liquids. हाँ, so ये yellow है, ये blue है. अब बताओ इसमें से कौन सा group लग रहा है ये? Don't look at this. Just look at this now. हाँ, ये वाला क्या है? First वाला B. Now let's break it down. थोड़ा सा रुको अभी मैं बताती हूँ. So here this is not showing clumping. This is clean, like there is no clumping over here. Here there is clumping. So this is showing clumping in B and no clumping in A. Therefore this is B. And this is a positive, RH positive and negative. So यहाँ पे भी clumping है. So this becomes B positive. So my blood group is B positive. Okay. You guys should also be positive. So here this one will therefore become O positive because here there is no clumping. दोनों में clumping नहीं है. AB अगर होता तो यहाँ दोनों में ऐसा clumping दिखता. Then it would become AB positive. Anybody has AB positive blood group? Anybody knows their blood group? So many people. So AB positive is known as universal recipient. Correct? Universal recipient. They can get blood from anywhere. Whereas O is called as universal donor. O cannot get from any other blood group. Only it can get from O. So this is how the problem of blood transfusion was solved by Karl Landsteiner. Let's go to the next part, which is regarding insulin. Okay, now we are talking about medicine. Remember, protein in medicine, protein in human health. What is insulin? Have you heard of the name insulin before? Injections लेते हैं insulin का? Yes? Diabetics के लिए, correct, very good. So insulin is actually a protein, it's a hormone in our body. What does it do? क्या करता है ये? It pushes the glucose from the blood into the cell. तुमको अगर बॉडी में एनर्जी चाहिए तो ग्लूकोज चाहिए. ग्लूकोज सेल के अंदर जाना चाहिए ना, लेकिन ब्लड में ग्लूकोज घूमता रहेगा तो इज इट ऑफ एनी यूज टू यू? यस, इट्स ऑफ नो यूज. द सेल हैज टू गेट इट, नो, इट हैज टू कम इनसाइड द सेल. तो सेल के अंदर जो इंसुलिन क्या काम करता है? ग्लूकोज को अंदर खींचता है, सेल के अंदर खींचता है. तो हमको जो एनर्जी मिलती है वो एनर्जी कहाँ से आती है? इंसुलिन की वजह से आती है. मतलब इंसुलिन का काम क्या है? ग्लूकोज अंदर खींचना. जैसे ही ग्लूकोज अंदर आ गया, आई विल लाइक हाँ आई एम फुल नाउ, मुझे खाना खाने की अब ज़रूरत नहीं है क्योंकि मेरे ब्लड में इनफ, मेरे सेल्स में इनफ ग्लूकोज आ गया है. सो आई विल स्टॉप ईटिंग. बट व्हेन देयर इज़ अ प्रॉब्लम विथ इंसुलिन इन माई बॉडी, जब इंसुलिन या तो कम होता है या फिर बिल्कुल बनता ही नहीं है, आई गेट अ डिजीज विच इज़ कॉल्ड एज डायबिटीज़. देयर आर 2 different types of diabetes, type 1 and type 2. बट उसको साइड में रखेंगे. बट डायबिटीज़ इज़ अ डेंजरस डिजीज़ बिकॉज इसमें क्या हो रहा है? मेरे सेल को ग्लूकोज मिल ही नहीं रहा है, इंसुलिन काम ही नहीं कर रहा है, या तो इंसुलिन बहुत कम है या फिर काम नहीं कर रहा है. इंसुलिन कौन करता है प्रोड्यूस? पैंक्रियास. देयर आर सेल्स इन द पैंक्रियास कॉल्ड आइलेट ऑफ लैंगरहैंस विच विल प्रोड्यूस दिस इंसुलिन विच इज़ अ प्रोटीन हार्मोन. ठीक है. सो इफ माई बॉडी इज नॉट प्रोड्यूसिंग इनफ इंसुलिन देन आई एम गोइंग टू गेट डायबिटिक. नाउ द प्रॉब्लम विद इंसुलिन also that this is the structure of insulin. So we all spoke about protein. अभी तक हमने बात करी. We said that antibody has a Y-shaped structure. Remember I showed you the picture. This insulin has a structure like this. This is how the insulin is when it is working, when it is active. Okay, when it gets denatured, what will happen to this structure? It will become a straight linear. Remember Sneha had shown you one, जब pH change हो गया, temperature change हो गया तो क्या हुआ? इसका structure change हो गया. So this is the actual structure of insulin as determined by scientists. In the early days, what was happening? People were having diabetes तभी भी. So what children, especially small children, were having diabetes. So what they would do? They have to get the insulin from animals. So they used to actually get pigs from slaughterhouses, pigs and calves. Okay, because that is very close to human insulin. But the problem was that from the pig or from the calf, they have to grind 2 tons of the pancreas. 2 tons. To get how much of insulin? Can you imagine? 2 tons of meat, of pancreas they're grinding to get 8 ounces, which is really 240 grams. Somewhere close to 240 grams of insulin. Grams. From tons, they're getting grams. So this was a big problem. So this is an early image of insulin, how it was produced by this Eli Lilly and company in the 1940s. Okay, and this was sold like that. So they had to take a lot of tissue, grind it up, purify it. They had to do a lot of steps to get insulin, and that was not enough for all the people who have diabetes, right, in the world. It was not enough. So what did people do then? क्या किया हमने फिर? क्या कर सकते हैं? Human का insulin gene लिया. What is gene? Part of DNA. So they took the gene from humans and they put it into bacteria called E. coli, Escherichia coli. Bacteria का advantage क्या है? Bacteria will multiply. फटाफट. It will go 2 to 4 in 20 minutes. Very good. Somebody knows the doubling time of bacteria also. In 20 minutes it will go, and so lot of copies of this insulin gene are then produced in the bacteria. ठीक है. So ये क्या किया मैंने? जीन को यहाँ से निकाला बॉडी से और वो बैक्टीरिया में डाला. ये कैसे किया? How was insulin made in bacteria like this? So you took the human insulin gene, you took one DNA of bacteria, this is a bacteria, DNA of bacteria which is called plasmid. We added this, it's literally like cut paste, okay, cut paste. यहाँ से cut किया, यहाँ पे डाला, paste किया. You put this gene, insulin gene with this plasmid back into the bacteria, वापस bacteria में डाला. Now this bacteria, is it the original bacterium? No, correct? Na? It has one extra gene. From where? From humans. The bacteria has a gene from humans. Then what we did, we put this bacteria and put it into a fermentation tank, big tank, because we want to produce lots of insulin, enough for all the diabetic people in the world, and also make it cheaper for them, right? So you put it into a tank. And then what do? Then you take the insulin out and then you purify the insulin. So insulin निकाला वो bacteria के cells को basically तोड़ दिया. You open the bacterial cell and you are getting the insulin. अब insulin को purify किया and you are selling it as medicine. So this is how the first time people put a gene from human being into a bacterium just so that we can get a large amount of this insulin produced. Okay, that can be used for human beings who have diabetes and that can be helping them. So this entire process is called as gene cloning. You must have heard of this word, gene cloning. So now what am I going to do? ये जो insulin gene है ये मुझे डाल दिया. अभी तुमको दिखाना है मुझे. ठीक है, यहाँ दिखाना है. All of you, I want to show you this. I don't have time to do this entire process, but I want to show you. तो मैं क्या करूँगी? If I show you insulin, तुमको कुछ दिखेगा आँखों से? नहीं दिखने वाला. So instead of that, what I am doing, I am going to take a protein which glows from this jellyfish. This jellyfish is called Aquaria victoria. Okay, it is found in American coast, Pacific Ocean, and this has this green color protein which will glow in the dark, or it is bioluminescent. Yes, bioluminescent. So you take this protein now, the gene for this protein, and what happens is when you put it under ultraviolet light, everybody knows ultraviolet light? It's bad for us, correct? So never stare directly at ultraviolet light. There is always some sort of protection. This is an ultraviolet tube light inside. So you always have some protection. You don't stare at it directly. Ultraviolet tube is harmful, but we will use it carefully to look at this fluorescence protein, this protein that is going to fluoresce. Now what we are doing is we are taking the gene from this GFP, that is the green fluorescence protein gene, and cloning it and putting it into bacteria. Again, the bacteria, does it have the GFP gene in its body? No. We are again doing cut, paste. We're taking the gene from the jellyfish and putting it into this bacteria and allowing the bacteria to grow, multiply. Lots of bacteria are growing. Then we do this in the laboratory. So there is another workshop that we do for BSc and MSc students, so basically college students, where they put this gene into the bacteria. They allow the bacteria to grow on this plate. So bacteria will grow like the small colonies. Can you see these dots? Those are bacterial colonies. Okay. So we actually do this entire gene cloning in our workshop, which is called as TA cloning workshop. This is a cloning workshop that we do at IISER Pune. At IISER Pune itself, we conduct this. So now let us look at this under ultraviolet light. The cloning has been done, but this is for you to see with your eyes now.
Okay, so we have these plates. You can see bacterial colonies on these plates. I don't know, one of this sample has the GFP gene cloned in it, and one kind of bacteria does not have the GFP gene cloned in it. But I cannot see it directly, right? I have to observe these plates under UV light, right? So this is called as a UV transilluminator. Now let's put them under the UV light. As she said, UV light is harmful. So we are going to have all this protection, and also we are going to see it from the top. Can we switch off the lights for this? Yeah, can you see this? So this plate, these bacteria have been cloned with the GFP gene from jellyfish, but these bacteria do not have the GFP gene. So under UV, these clones appear green in color, they glow as compared to the other bacterial cells. Any questions? We'll also show you one more thing. These bacterial cells are growing on solid medium on a plate. We also have certain cells growing in liquid medium, right? So let's see how that looks. Can you see the glow? See, the bacterial cells are at the bottom of the tube, not in the entire tube. The cells are at the bottom of the tube. Are they glowing? So are we successful in putting a jellyfish gene into a bacteria? Are we successful?
So what all did we learn today? Proteins are basically big molecules which are folded, okay? And that is how they get their activity. So how did we know activity? What did we look for activity? The different frothing of potato extract with hydrogen peroxide. That showed us the activity of a protein. We looked at that. Then what did we look at? We saw the different types of proteins. We discussed that. What are the different types? Some of them we actually saw. So you could see the insulin we showed you as a structure. The antibody antigen reaction also we showed you. One of the enzymes, catalase, we have shown you. We have shown you one gene which is the glowing protein, that is GFP, right? That was another kind of protein that we have shown you. We have also discussed how under different conditions of pH, temperature, you will have a folded protein versus a inactive protein, or a what Sneha said, it is broken protein, a protein that is broken at different pH and temperature conditions. Then we actually saw some of the applications in medicine, in human beings, in medicine, how the proteins can be used. You also know that the wool and silk, that is textiles that are produced, they are also kind of proteins, right? I hope you know this. This is coming from animals, but silk is coming from which silkworm, and wool is coming from sheep. But both of these are also kind of proteins. You also have proteins in testing, diagnostics. Everybody knows the RT-PCR test and then other tests that you did. What was this test called? Rapid antigen, RAT. Have you heard of this test during COVID? Right? Then we saw some recombinant protein example like insulin, which was produced in bacteria, in a different organism. Finally, what is our conclusion? What is our conclusion? Proteins are lifting life up, making life better, making us strong. Yes, people who do a lot of exercising, they get muscles because of proteins, right? Building up of proteins. So with this, thank you for your patience. Thank you for staying through the entire session.
बैक्टीरिया ग्लो क्यों करते हैं?
Bacteria glow क्यों करते हैं? Okay. So the bacteria that we used does not naturally glow. We put a gene inside the bacteria, as ma'am said, cloning use करके jellyfish से gene जो glow होता है वो produce करता है protein, वो bacteria में हमने डाल दिया. That's why the bacteria can glow. The other bacteria, was it glowing? Was it glowing? No, because it did not have that gene. But in nature, you have a lot of bacteria called bioluminescent bacteria which naturally produce these glowing proteins. So those will be those kind of bacteria. The one that we used, it does not naturally produce it.
I didn't understand the protein storage function.
Storage.
Okay, so proteins are, I told you about the 3D structure that they have. They are long chains and they fold amongst themselves and they form a 3D structure. So within that 3D structure, like a cage-like structure, they store the iron molecule. So the structure of that ferritin protein is such कि वो iron को trap कर लेता है. That is how it carries the storage function. और जब iron छोड़ना होता है, it will change its structure slightly and the iron will be released. Similarly, you have many other proteins which do storage jobs. Is that clear? The protein helps to catch that molecule and store it.
Ma'am, I did not understand how they add GFP gene into bacteria and bacteria becomes the insulin.
Bacteria is not going to give insulin if you're putting GFP. GFP we have added so you can see it. If I put insulin gene in bacteria, I can do it. But will you be able to see anything under any light? Nothing. You won't be able to see whether the gene has gone or not until you produce insulin and actually inject it into a person who has diabetes, and then you'll figure out that the insulin has actually worked. But with GFP, what was the advantage? We could see it with our eyes. So it was an example where instead of putting insulin gene, we have put GFP gene into the bacteria so that we can see that the gene has gone into the bacteria and it is actually giving the protein. It is expressing the protein. So the protein can be seen with our eyes. That is why we did that. GFP and insulin are two different things. They are not the same.
Experiments. They are two separate experiments. Last question. Sorry.
Okay, so ma'am, the guy who found out the blood groups,
Yes.
how did he get the idea?
The Einsteiner, how did he get the idea? How did he figure it out?
The Nobel Prize guy.
How did he figure out what? That the blood group is present?
Yeah, how did you get the idea?
I don't know the exact methodology what he used. I really don't know what he did.
He was working for many years and that's
He published a paper which said that antigen antibody reaction which causes the agglutination.
So basically what was happening was that the primary observation was that in many cases the blood transfusion were working and in many cases the patients were dying. Okay, so that's when he figured out that there are subtypes in the blood, and those subtypes he then, through careful experimentation, he figured out which subtype can give to which type, and then once that got figured out, then he kind of back calculated the stuff. Okay, but precisely what his methodology was, how he ended up doing it, honestly speaking, I have no idea about that. Okay, basically what we were trying to show you over here, people, if we can have your attention please. What we were trying to show you over here is that there are lots of biomolecules which make up our body, and not only our body, every living organism, their body is made up of those molecules. And these molecules, they have their own properties. And if you understand those properties, then you can understand how you can use those properties for human benefit. I mean, it's obviously interesting and important in their own right to understand them, but at the same time, if you appreciate that, then you can use it for human benefit. So for example, if you first understand about protein, then you understand that the proteins, they are not going to function at all times, right? When you understand that proteins are not going to work all the time, if you change the condition, then proteins will stop working or they will become less active. Then you understand that things that are happening around us, they will end up affecting the proteins. And once you understand that, then you understand that because we can manipulate the proteins, now it is possible for us to change the proteins or change the conditions in such a way that even under climate change, we will be able to have those organisms, those plants, those animals, etc. still being functional. Okay, so that was one importance. And the other important thing that we were trying to show you was that how exactly can one take the proteins from, or rather the genes from one organism to another, and thereby make a protein which was not being made by an organism now getting expressed in that organism. Okay, so that's where the insulin example comes in. You take the insulin gene from humans and then you put it in bacteria, and once you do that, you are able to get insulin in very high amount at a very cheap cost, which actually saves millions and millions of lives every year on this planet. Okay, so that is the importance of this technique called cloning. We did not show you cloning, but we showed you a result of it. And if you can see right now what is there, you know what that is? That is just molecular biology written, but what is it? The same GFP expressing bacteria that we showed you as colonies, we took that on the plate and we wrote molecular biology with that, and that is glowing under ultraviolet light. Okay, so this is just a way of showing that sometimes you can even take these things and make cool aesthetic art out of it. Okay, that's about it. Thank you very much. Thanks for coming. Ashok, over to you.
अभी देखिए इतना बढ़िया टॉक, तो हम सभी अभी प्रोफेसर सुतीर्थ, स्नेहा एंड पूर्णिमा का बिल्कुल जोर से ताली बजा के थैंक यू बोलेंगे, एंड थैंक यू वेरी मच टू यू ऑल. ओके.